All right. Hi, everyone. Welcome to another episode of Breakthrough. Breakthroughs with us? No, Breakthrough. Breakthrough with Sam and Tiff.
I mean, it's up to... We can change it if you want. I don't know. I don't know which is better.
So we changed the name of the podcast from Sam and Tiff Talk Science to Breakthrough because we just thought it was catchier and more simple.
Yeah.
And more relevant.
Also, I think people find, if you say science, I think it's very fascinating. But some people, shockingly, find it boring.
Wow.
Shockingly.
I know, we have to get out of our little science bubble.
I know, it's weird, right? It's like people don't love science. That's crazy.
How can you not love science? Anyways, I'm your co-host, Tiffany. I did my PhD in oncology at the University of Oxford, where I met Sam. And now I'm part of two biotech startups. I co-founded Gambit Bio, where we're using saliva to detect cancer earlier through home self-test kits. And I'm also a senior scientist at Infinitope's Cancer Vaccines, where we're making cancer vaccines.
Super cool. I'm Sam. I'm a doctor.
Is that it?
Yes.
Oh,
short and snappy. That's what you want, no? Short and snappy?
You need to make a one-liner for me.
For you?
Yeah.
Yours is good, though. Yours is very... I like yours.
All right.
A bit more detailed than mine.
You also learn about the cultural differences between Americans and Brits. Anyways, I can't believe it's November. So we're going to talk about...
It's been a crazy year, right?
Yeah, it's been a really crazy year. We had a bit of a break because we were both very busy, but we're back now with the 3 breakthroughs from October. And yeah, it's been quite exciting in both my world of cancer detection and in the world of malaria, which we'll talk about later. But Sam, do you want to start us off with what's happening with Grail, the major cancer detection company?
Sure. So, so Grail is a company in California in the US, and they have probably, I think it's open to interpretation, but probably the most exciting multi-cancer early detection kit on the market currently. Would you think that's a fair?
That's very fair. They probably have the biggest amount of money raised for a liquid biopsy cancer detection test.
How much have they raised?
A ridiculous amount, in the billions, and they're
really, wow.
Their valuation is also in the billions.
They're public, aren't they?
Yeah, exactly. And they've been around for years, but a liquid biopsy is basically you take your blood and then you look at the biomarkers inside the blood. And what they're famous for is making circulating tumor DNA, also sometimes referred to as cell-free DNA, more mainstream in the commercial context of cancer detection tests. And they look at the methylation of the circulating DNA from the tumors. And yeah, they do something called methylation sequencing. And they've had some previous clinical trials before, but now they have done newer trials, which, you caught from the ESMO announcement. So ESMO is another huge conference in the oncology world. It stands for the European Society for Medical Oncology. And it happens every year, and they decided to announce some really exciting interim results.
Yeah, and they did a huge study. So they recruited about 25,000 adults, asymptomatic adults, and they gave them all the Galleri early cancer detection test, and then they followed them for a year. So it's a blood test. Looks at the cell-free DNA, as you said, and it gives you 2 indications. So first, is there a signal of cancer? Second, if there is, it gives you an idea of what the tissue of origin might be. And they are able to detect 50 plus cancers, including all of the kind of most common solid cancers.
As well as several of the liquid cancers, the blood cancers. And what's really impactful, the takeaway is that they can detect cancers for which there are currently no standardized screening guidelines, including for things like head and neck and pancreatic. And usually you don't see the symptoms come up until it's too late. So that's a huge deal there. And the second takeaway was that their PPV
Yes.
a positive predictive value, which is different to, measurements like sensitivity and specificity, is 62%, which is actually so ridiculously high
So that is
and shocking, especially to a clinician, right? Like, should we talk about what PPV is?
Yeah,
first?
100%. So the number 62% doesn't sound crazy, right?
It doesn't sound impressive.
That's an insane number. So to put it in context, right? So you can explain what PPV is, but just to put it in context quickly. So there's another test called TrueCheck, which is a similar multi-cancer radio detection test. And their PPV, do you know what it is? By any chance, I can tell you. It's
much lower.
It's in the same asymptomatic population. That's really important, by the way. It's asymptomatic, not symptomatic. Asymptomatic is 3.5%. 3.5%.
Wow.
This one, Galleri, 62%. So
And was it also a multi-cancer detection?
Yes. So tell us what PPV is, because it's also an important concept.
Yeah. So obviously people know sensitivity, how many true positives you can get, and then specificity, how many true negatives you can get from a certain test that you're using. But those values are limited to the test itself. It doesn't measure it in the context of a true screening population in the real world. And it doesn't take into account the prevalence of the disease that you're trying to screen for, whereas PPV takes that into account.
Yeah, very
so
important.
Positive predictive value, PPV, basically says if you get a positive results from this test, what are the chances that it's actually positive? And 62% is huge. It's really orders of magnitude higher than what other test kits or like current screening techniques would give you.
So if I take a TrueCheck test now, right, and it's positive, the chances that I actually have cancer is about 3.5%. So almost every single positive test is a false positive. Almost every single one. In this case, if I take the test and it's positive, my chance
more than half of
is
them
62%. It's very, very high. So
like
really,
2/3 of them.
It really is impressive data.
It's really impressive. It makes the clinicians, like a clinician like you, trust the result of the test more because you're like, okay, there's very high chance that we do have to have a clinical follow-up.
Yeah.
And they measured that as well. They measured how long it took from a positive result to an actual clinical follow-up. And I think the average amount of time was like 30-something days for a positive result.
Pretty fast.
Yeah, exactly. So like from the moment you get a positive result from a Galleri test to the moment you do something about it, it's 30-something days.
And so this doesn't confirm cancer, right? So like if let's imagine I take the test, it's positive, what happens next?
You might get an MRI or a mammogram. So basically the whatever is the gold standard in the clinic for that specific type of cancer. And this brings me to a good point. They're also able to relatively accurately determine where the cancer is coming from just by looking at the methylation patterns from the DNA.
Very exciting.
Which is really cool because, what's the point of knowing you have cancer if you don't know which organ to focus on?
And that's a major problem as well. So I know we're talking about asymptomatic screening here, but in the context of symptomatic patients, sometimes you have a symptomatic patient, you're strongly suspecting cancer.
They have like general symptoms.
Exactly.
Like weight loss or...
Exactly. And even if you do a full body scan, like a CT, chest, abdo, pelvis, for example, even if you do that, often it doesn't give you the answer. It doesn't tell you where the cancer is. So if you had a test like this, that would be really useful for that as well.
Exactly. So it really takes out the guesswork and the clinical pathway is less complicated, less costly. So it's a great test. But my only big caveat is, of course, I run early cancer detection company myself. One of the caveats is accessibility. How affordable is your test kit? Because the last time I checked, it was around $1,000 for whatever is commercially available right now. So, that's not going to be very affordable.
100%. So
and how often can you screen for this?
Exactly. It's for quite a specialist market, right? If it's $1,000, clearly it's not for everyone.
Yeah, exactly. And our insurer is going to cover it if you're in the States. And how is, a system like the NHS going to afford it?
Exactly, 100%. And they would have to prove the cost effectiveness. And by the way, that's the next step that they are doing. So they are moving.
Oh yeah, how are they doing that?
You mean the next step? with the NHS?
Yeah, I think
they're running a massive trial in the NHS. So they're taking it's hundreds of thousands of patients who are already heading to asymptomatic screening for breast, colorectal, cervical. So the existing
That's why
pathways.
we should clarify that this Galleri study has two parts: one in the U.S. with 25,000 people, and then a much bigger one in the UK with the NHS.
That's not read out yet, though. The US study is the one that has read out with the numbers we've given.
Exactly.
The other one, the NHS one, is
ongoing.
Yeah.
So yeah, keep an eye out for this space. But that would be my only big red flag would be, even if the results are great, how many people can afford it? And does it actually help a system like the NHS where, the current setup is that like the NHS pays for everything?
Yeah.
Yeah.
Exactly. Super cool.
But yeah, super exciting. We're moving towards an era of preventative medicine and early detection.
100%.
It's where the future's at.
It's very exciting.
Yeah. Awesome. I love ESMO and ASCO, which is the American version of ESMO.
Yeah. Always the super exciting things getting
where Sam gets his exciting news. So the next breakthrough from the past month is on one of Sam's favorite topics, malaria.
So we've spoken about this before.
A few months ago, wasn't it?
We're kind of at the forefront of malaria. We've talked about malaria before. We talked about the anti-parasite nets.
Exactly.
That's what we talked about before.
So a few episodes ago, Sam and I talked about how these Harvard scientists they created these mosquito nets that are novel and it helped reduce the rates of malaria infections or transmissions.
Yeah.
But now there is another breakthrough in the same year.
There's so many. The malaria breakthroughs are amazing, and we'll give like a bit of the story because there's still work to be done, clearly. But the breakthroughs are so impressive. So malaria is still a disease that kills about 600,000 people a year.
That's really tragic, and it's 2025, and this is still a huge unmet clinical need. And most of the people affected are kids.
100%.
Children under the age of 5.
Most are less than 5. And most are in sub-Saharan Africa. So it's very much it's actually very interesting because malaria concentrates in the places where there's also sickle cell disease, and that's because of coevolution. So sickle cell disease is protective against malaria. So that's why sickle cell disease actually continue to evolve, because that's where malaria is. It's very interesting kind of coevolution.
Yeah, it's kind of like
that's why it's in.
It is really cool because it's kind of like a way for you to combat malaria.
Exactly, exactly. It's very interesting.
But what's interesting from the past month is that the giant Novartis which is already an expert in anti-malarial drugs, has come up with a novel treatment that has met a phase 3 endpoint and has basically beaten the gold standard on market right now, which they made, by the way, I think.
Yeah.
And this new drug is called GanLum.
GanLum.
And I'm going to hand over to Sam to explain what that is.
So, and the headline is that the 97.4% of the patients in the phase 3 trial were cured. 97.4%. So that's very major numbers.
And this is taking into account like failure to adherence to
yes, exactly. So we're taking the conservative number. If you take the liberal number, it's like over 99%.
Exactly. So this already takes into account things that are unrelated to the trial, like a patient passed away, or it's not effective because of something outside of the trial.
Yeah.
So that's a really conservative number already. So huge success.
So the major issues really, so we do have good malaria drugs, but there are two major issues. One, resistance. Two, it doesn't block transmission. So they're called artemisinin.
I could not pronounce that word.
and because we've been using them for so long, the parasites that cause malaria have developed slowly resistance to the artemisinins, which makes a lot of sense, right?
Evolution.
So, exactly. So this drug, GanLum, has an completely new mechanism of action. By the way, discovered beautifully by Novartis. They screened 2.5 million
compounds
to search for anti-malarial activity. And the top hit was GanLum. Actually, it was one molecule called ganaplaside.
Yeah, there you go. And the lum part is it's an existing drug.
Exactly.
But they have two mechanisms of action.
Yeah.
The gan part is the novel one.
Yes.
And apparently it disrupts the protein transport system.
Exactly.
That happens within the parasite. Whereas the lum part, I'm trying to see what it stands for. Is that one the original mechanism of action?
So that's used already in the existing, it's called lumefantrine. That's already used in existing malaria treatments. But
exactly.
So because it's got an entirely new mechanism of action, it clearly isn't inhibited by the existing underlying artemisinin resistance. That's the first thing that's really exciting, beyond the 97.4% cure rate. The second thing is that it actually could and should block transmission because the existing artemisinins do not target the parasite, the malaria parasite, when it's actually in the sexual stages. So when it's actually causing reproduction and leading to more parasites. Whereas GanLum does target the sexual stages and therefore probably, I don't think we have explicit evidence, but probably blocks transmission too, which is critical.
It's so smart, and it's two-pronged approach because the previous drugs, they might cure the patient of the symptoms, but if a mosquito comes and bites the patient, they transfer the parasite that's not mature yet, but still at the sexual development stages. And the mosquito then transmits it to another person.
Exactly.
But now it's killing the parasite at those early stages of development. So that's great. That's prevention and cure in the same drug.
Exactly. So it's like a triple hit. It's more effective, and it blocks transmission, and it overcomes resistance. So that's why it's such a big deal.
It's such a big deal. And they said it's a first big innovation in anti-malarial space since 1999.
Yeah.
So in a quarter of a century.
It's been a while.
Yeah.
In terms of just a bit of context, and we discussed this last time as well, but at the turn of the century, since the turn of the century, the number of deaths from malaria have been cut in half. They've dropped by half, which is really good. And now with innovations like this and with a number of others, we should be able to move slowly towards malaria eradication. And there are lots of other breakthroughs this year. So there's now 2 approved vaccines. There's lots of other vaccines coming through in the pipeline. We now have new anti-malarials.
And mosquito nets.
Mosquito nets, anti-insecticide nets, anti-parasite nets, things called spatial repellents, which get rid of the insects, the mosquitoes, and kits and preventative prophylactic drugs as well. So in other words, there's loads of things that are happening.
Yeah, I think we're going to see the
deaths
eradication in our lifetimes.
I hope so. So some countries, so China is a good example of a country that did have malaria and now has officially eradicated it. So it is possible to do it.
Absolutely possible. And we're definitely moving towards that. And now it's just a matter of logistics and integration into the healthcare system.
Yeah, exactly. Access is the major issue. So this drug is not quite approved yet. It's probably going to get approved in the next 12 to 18 months. Mm-hmm. When it does, access is the major issue. So clearly price is important. And then how do we actually distribute the drug effectively?
You say it's easy to take it because apparently it's a daily sachet for 3 days and that's it.
Yeah. So apparently it tastes really disgusting. Oh, so one of the major issues in the trial was vomiting, but not because it interferes with the GI tract or anything, but just because it tastes disgusting.
It tastes so bad. Can they just make it a cough drop kind of thing?
Yeah, exactly. So you can change the taste of things, right? So it's not an insurmountable problem that it tastes disgusting.
Yeah, that should be fixable, I think.
Exactly. And there are lots of existing drugs, by the way, that we use that do taste really bad. We use them all the time in the clinic. And
you think Big Pharma would spend more money into making it taste good?
they should do, right? So, but that's an important kind of.
They're we already spend money on screening 2.5 million compounds.
Yeah. This is the last problem. How do you make it not taste disgusting?
That should be relatively easy.
It should be. It shouldn't be as hard as the massive screen, right?
Exactly, but. Wow, what a Herculean effort to screen all those compounds and. Yeah, good on Novartis and very exciting.
100%. Did an awesome job.
Yeah. Awesome. Cool. So for the third breakthrough, would you like to talk about this? I think you got quite a lot of attention on Twitter or Instagram from sharing this news.
So this is a nice, simple story. It's the first ever AI-enabled pregnancy.
Crazy. And it's just one pregnancy.
One pregnancy. So it's offered at the Columbia University Fertility Centre. It's the only place in the world currently that offers this approach. And they recently just now had the first ever pregnancy via this approach. They published it in the Lancet. So the
super impressive, and they call it the STAR system, which stands for Sperm Tracking and Recovery System.
Yeah.
And it's a combination.
It's a super simple idea.
Yeah.
Like, beautiful use of AI, I think.
It is beautiful because it combines it with hardware and imaging. So it's not purely a software solution, but it combines like microfluidics with imaging, with analysis. And I think this is one of the best parts to use AI in.
100%. So the major problem is, so it's about male factor infertility. So,
which actually accounts for 40% of infertility issues. And sometimes people don't think about that.
Yeah, 100%. Yeah. And one of the underlying causes of male factor infertility is azoospermia. So it's very few or zero, apparently zero sperm.
And this is in around like 10% of the cases.
Of the 40%, yes.
Yeah, exactly.
And at the moment, if you're a man with azoospermia, then what happens is you have a fertility scientist look through the images of the sperm, trying to find individual motile sperm, right? That's how
you
it
literally
works.
have like an embryologist looking at a slide with your potential sperm on it for like hours and hours, trying to look for a functioning, swimming motile sperm.
Exactly. And inevitably, that doesn't work very well, right? So
it's not scalable. It takes forever. It's subject to human error.
Exactly.
If I'm tired and I haven't had my coffee that day, I might miss like a motile sperm
100%.
under the microscope. But
whereas this is an AI platform, and it screened two. So this is a couple that had been infertile for about 19 years. They'd been trying for 19 years.
It's crazy. And they're both like late 30s at this stage.
Exactly. And then they got to this approach, and the AI platform screens through 2.5 million images
per hour,
looking for a motile sperm, and it found two. In these 2.5 million images, so of course the humans miss it, but the AI finds these two motile sperm. You can then grab one of them and use IVF, so it's an intracytoplasmic sperm injection is the approach. And that led to this pregnancy in the couple who'd been infertile for almost 20 years.
And they've tried so much. Both parties have gone through multiple surgeries or biopsy testing, which are very painful, and they can end up with, as you just alluded to, like no functioning sperm being identified. And it's really disheartening, and it's a very sad, situation that a lot of couples face right now, even though it might not be as extreme as like 20 years of not having any successful pregnancies. But apparently this couple, they've had one motile sperm found before, but the implantation or something didn't work. So this was like literally their last hope. And the woman also had her own issues with her ovaries. So it was like probably like one of the hardest cases to solve that they found. And they made it work. So it's a great proof of concept in the first couple. And like you said, the human didn't find any sperm, but the STAR system found 7 sperm, 5 of which were not swimming, so you don't want those. And then 2 of them ended up being the healthy ones.
Yeah.
Yeah.
So there you go. Really amazing use of AI. And as I said, it's only available currently in the Columbia University Fertility Centre.
Yeah.
But presumably this will eventually be available around the world for, other people to benefit from.
Yeah, it's really smart. And they basically, apart from being able to like screen like more than something million images.
2.5 million.
2.5 million images per hour. They also have 300 frames per second, I think. So it's like the camera, the imaging technology is also a huge part of it. And same as the microfluidics, because like once the star system identifies a motile sperm, or even just any sperm, like the non-motile ones. It traps it with like a bubble, and then it encapsulates it in a little chamber that you can then take to do the rest of the procedure. Super cool.
Super smart, right?
Yeah.
It's like simple, smart, very useful.
Yeah, exactly. Honestly, like one of the best applications of AI in
100%.
science.
Yeah, you can't really argue against that, can you?
Yeah, exactly. And it's so you just need to train it on lots of images so that it doesn't pick up a speck of dust instead of like a sperm.
Right.
And what they do is they also follow the sperm. So they have to be able to identify that sperm in multiple time frames. So then you know for sure it's not a speck of dust because you've seen it in multiple time frames. It's so smart.
Super smart. Super
Yeah.
nice.
Super cool. So yeah. And huge impact because don't like 1 out of 4 couples have infertility issues.
Yeah.
So huge market, huge unmet need, and yeah, AI really is helping. So when that baby is born, I guess it'll be the first AI enabled
Baby.
Oh gosh.
Yeah, weird concept, but there you go.
Awesome.
Good.
Cool. Well, really cool month.
Super
And
cool.
we're hurtling towards the end of 2025. So yeah, come back next time for more breakthroughs from the end of the year.
Awesome. Short and snappy.